right, so good morning. Happy Thursday or Friday. What you guys are going to work on today is the beginning part of the lab that we'll do in class next week. So you're going to do the knowledge probe for transformation lab, which is going to use bacteria. And actually what you're going to do is change the genes of the bacteria. We're going to make it so that it will glow. And then you'll also, underneath the black light, you're also going to look at the fly and do some research on the genes of your flies so that you can get to know what to predict for your phenotypes. And then there's a knowledge probe just on bacteria a little bit there on this idea of transformation. So let's take a look at the lab real quick. When you open it up, it'll look like this. So you're doing bacteria transformation. And these first 4 questions are just on bacteria in general. You will probably get a little bit of, you might have to look at this one. I think if you read through here, it also talks about it too. But bacteria transformation essentially is when bacteria take up loose DNA out of their environment. So they take in this information and then they incorporate that DNA. They take in DNA, loose pieces of DNA, and then they incorporate that into their plasmids. Okay, so it's a way for them to get some new DNA and to get some genetic variation. You'll need to read through this information, and then there's some other questions that come up. This gives you some background on how the transformation process will occur and what you're going to do. You'll go through the activity. This gives you background information about the fluorescent protein that we're going to insert into our bacteria to make them to glow. And along with that, we're also going to give them the antibiotic resistance, which is how we'll know that they have taken up that gene, because we'll grow them on plates that have ampicillin on them. The gene itself is activated by arabinose. So it's a gene that is regulated by a operon. Okay, and so we'll talk about operons more with gene regulation, but essentially what's going to happen is that if arabinose is present, they make these proteins to make the gene that glows. If arabinose is not present, then they won't make those genes. They won't transcribe them to make that protein. So depending on what information is present will depend on what they do. This is a diagram of the plasmid that we will actually insert into the bacteria. A plasmid is a small piece of DNA that's in bacteria cells. It's a small round piece besides their main DNA, which is the bigger circular piece. So on our plasmid, there will be the gene for ampicillin resistance, the gene for the glow protein, and then the gene that is arabinose dependent for expression. So this part will only get transcribed if arabinose is there, because then that transcription activates the transcription of the glowing, where the ampicillin resistance is going to always be transcribed when the cell is doing that. There's a really nice video that talks about how the GFP protein got put into this plasmid. It's a protein that's found in jellyfish and has been used widely in biology to see to be able to track genes. So it's kind of a quite interesting little video there. All right, and then we will start with the prediction when in class on Monday or Tuesday, depending on what day you are doing the lab. So that's what you're going to work on for this. For the flies, you're going to read through this first knowledge probe that talks about how to identify a male versus a female fly. These are both wild type, where you have the red eyes, the normal wings. The males have the blacker abdomens versus the females have the lighter abdomens. And then they also have sex combs, which are not, oh, these arrows are off. The sex combs, oops, are these little tiny black, I guess I moved my arrow here. The little tiny black hairs that are in this circle. Okay, so see how there's that circle there? The sex combs are those. And so for me, that's what makes it easier to see where, whether it's male or female. I like looking for that. The females don't have those sex combs. But you can also look at the abdomens. We're not doing any that are black body versus yellow body, so that shouldn't be a problem. Sometimes we run into problems with that. But what I would like you to do is to work on the prediction, recording the traits that your P generation is. That's what's labeled on your vial. That's the P generation. And then what you're going to do is do some research on that, figure out, is your Apterus dominant recessive, is your white eye dominant recessive, is your sepia eye dominant recessive, and then the chromosome number it's found on. From there then, if it's a sex-linked trait, it'll be on chromosome number 1. If it's not sex-linked, it'll be on chromosomes 2, 3, or 4. The fruit flies only have 4 chromosomes. And then you're going to make a Punnett square to predict what you think your F1s should be. Those are the flies that are in your vials right now. And then you'll also make a prediction for your F2. So when your F1s are crossing, what kind of babies are you going to get? So the larvae that hatch out, what kind of flies do you predict to get there? Okay. And then you also have the dihybrid that you're doing with your partner, which is the Apterus and the sepia. So you'll do another prediction for both the F1 and the F2 offspring doing a dihybrid cross. Okay. So that's what I would like you to work on today. And if you have questions, always shoot me an email. Otherwise, I hope you have a great day, and I'll see you the next time in class.